Good morning, class. In our previous class, we had discussed mitosis and its stages in detail. Today, we will be talking about meiosis. Meiosis is that kind of cell division which occurs in the reproductive cells of both the male and the female, and it occurs at the time of formation of gametes, what we call as gametogenesis, in plants as well as animals. And during meiosis, a diploid cell having 2n number of chromosomes undergoes two successive divisions, one after the other, to give rise to 4 cells, and these 4 cells are haploid. They have n number of chromosomes, or we can say that the cells that are formed, the gametes that are formed, that is the male gamete called the sperm and the female gamete called the ovum, contain only half the number of original chromosomes, and because of this, meiosis is also called reductional division, because the chromosome number is being reduced to half, from 2n to n number of chromosomes. Now this is a very important concept which each one of you should understand, that why does meiosis occur in male and female reproductive cells at the time of formation of gametes. Now to explain this, I will say that all sexually reproductive organisms, those who reproduce sexually, their body cells contain, the somatic cells contain a diploid number of chromosomes, and it's only the gametes that contain haploid number of chromosomes. Gametes, that is the egg cells and the sperm cells, they cannot have diploid number of chromosomes, because imagine if that happens, then at the time of fertilization, when the fusion of the male and the female gamete occurs for the formation of the zygote that will be produced will have 4n number of chromosomes if reduction division or if meiosis does not occur and the chromosome number does not become half, that is n. So in that case, what will happen? 2n number of chromosomes from male and 2n number of chromosomes from the female will fuse to form a zygote having 4n number of chromosomes. Now to explain this again, I will say every body cell of a human being has 46 chromosomes, and if egg cell and sperm cell also have 46 chromosome, what will happen? At the time of fertilization, the zygote produced will have 46 plus 46, which makes it 92 chromosomes, and the offspring that is produced will have 92 chromosomes in each of its body cell, because after the formation of the zygote, only mitosis takes place, and mitosis, the same chromosome number is maintained. So every cell of the of such an organism which has, or such a human which has 92 chromosomes in the zygote, every cell will have 92 chromosomes, and if meiosis does not occur, then the chromosome number will keep on doubling with each successive generation. The next one will have 92 plus 92, that is 184 chromosomes. So this is not possible, this does not happen, and gametes therefore contain only half or haploid number of chromosomes. And when the haploid gametes fuse, the resulting zygote that is formed will get a full set of diploid number, and you know the chromosome number for a species is constant. It is specific. It does not change. All humans all over the world will just have 46 chromosomes in their body cells. So meiosis helps in the formation of gametes containing haploid number of chromosomes, and by the fusion of such gametes the constant chromosome number, that is 46, is maintained in humans, and we can say a constant chromosome number is maintained in every species. Now, as I told you, that meiosis involves two successive divisions, meiosis 1 and meiosis 2, and the various stages of meiosis I have just listed them here. So you can see that before cell division occurs, the cell enters a resting stage which is called interphase. Then meiosis 1 takes place, which is actually the reductional division, and here the chromosome number will become half. And as I have already told you in mitosis, while teaching mitosis, revising mitosis with you, that karyokinesis occurs, and there are 5 stages of division of nucleus, that is prophase 1, metaphase 1, anaphase 1, telophase 1. These 4 stages are there for the division of the nucleus, and prophase 1 is the longest and is further divided into 5 substages. Here lies the difference between mitosis and meiosis. I will now explain each of these 5 stages of prophase 1, which are liptoten, zygotene, pachytene, diploten and diakinesis. And after these 5 stages of prophase, it is followed by metaphase 1, anaphase 1, telophase 1, and then there is a short interphase 2, which is followed by meiosis 2. And meiosis 2 is the second division which takes place, and this division is like mitosis. We call it equational division, because after meiosis 1, chromosome number has been reduced to half, and then 2 cells that are formed from a diploid cell will have half number of chromosomes. They undergo meiosis 2, second division, which is equational in nature, and each of those 2 haploid cells will form 2 more haploid cells like mitosis, and at the end of meiosis 1 and meiosis 2 we have 4 haploid gametes. And in meiosis 2 again karyokinesis is divided into 4 stages, or these are prophase 2, metaphase 2, anaphase 2 and telophase 2. So we will begin with the different stages of meiosis that have been listed here, and we will be doing each of these in detail, one after the other. So let us begin with meiosis 1. Now meiosis 1, it starts with the cell entering in the resting stage or the stage of growth, which is called interphase, because though the cell appears to be resting, it is physiologically the most active stage, interphase. Here the nuclear membrane remains intact, the nucleoli are prominent, and DNA, important point, DNA gets replicated, and chromosomes are in the form of chromatids. They appear as a pair of chromatids. which were actually present as a network called the chromatin network. Then the cell enters the dividing phase and meiosis 1 takes place. Prophase 1, as I told you, is divided into 5 substages. So the first stage is liptoten. Now each of these stages, I have mentioned the important events that take place here in points and the diagrams for each of these substages. So here I would say that you should be able to understand these, and for that read from the book also, write the important points, draw the diagrams, because this is something new that is being taught to you this year. You have not studied this before. So prophase 1, as I told you earlier, is divided into 5 substages. The first is called liptoten, and in liptoten the chromosomes are shortened and they are visible as single strands. Second point, there is beaded appearance of chromosomes because of the presence of chromomeres. The nuclear membrane and nucleoli are intact. Then the cell enters into the second stage of prophase, which is called zygote. Zygote is that stage in which the homologous chromosomes, that is one set of chromosomes that are maternal and the other that are paternal, they will pair up, they will come together. The paired homologous chromosomes are then seen as bivalents, that is, 2 chromosomes that are coming together, and later each of these chromosomes will be divided into 2 chromatids. So 2 chromosomes with 2 chromatids each, they are also called tetrads. That is, you can see 4 chromatids in a bivalent. So the pairing process that takes place is called synapsis, and this synapsis is accompanied by the formation of a complex structure which is called synaptonemal complex. And there is thickening and shortening of chromosomes. The nucleoli, they tend to disappear. Now we have studied about leptoten and zygote, and these 2 stages are of short duration, and the next stage, that is pachytene, is longer. And now we come to the characteristic events that occur in pachytene. So pachytene is an important stage and a longer stage of karyokinesis, prophase 1 in meiosis. Now pachytene is characterized by the appearance of recombination nodules, which are the sites where crossing over occurs between the non-sister chromatids of a homologous pair. I hope you follow the term non-sister. Non-sister means chromatids of 2 different chromosomes. In homologous chromosomes, I told you, one is from the mother, one is from the father. We call it the maternal set and the paternal set. So crossing over takes place between chromatids of maternal and paternal chromosome, not between the chromatids of only maternal or only paternal. One maternal and one paternal chromosome, as you can see in the diagram also, that chromosomes of a homologous pair, which were called bivalent chromosomes, they become twisted spirally around each other and they appear as tetrads. Crossing over is actually the exchange of genetic material between the 2 homologous chromosomes, and it is an enzyme mediated process. The enzyme that is involved for crossing over is termed recombinase. Then exchange of corresponding chromatid segments take place and the nucleoli disappear. Now, recombination between the homologous chromosomes is completed by the end of this stage, that is by the end of Pachytene, leaving the chromosomes linked at sites of crossing over, and Pachytene is followed by Diplotin. Now, Diplotin is recognized by the dissolution of the synaptonemal complex, and the homologous chromosomes of the bivalents separate from each other except at the places where crossing over has occurred. Now, the X-shaped structures, they are called chiasmata, and the bivalents will repel each other. The chromosomes that are joined at several points along their length called chiasmata, and chiasmata are the sites of crossing over. So I have listed these three important points under Diplotin that occur during meiosis 1, prophase 1 of meiosis. Diplotin is followed by the last stage of prophase 1, which is called diakinesis, and diakinesis is the final stage of meiotic prophase 1. The chromosomes are fully condensed and the meiotic spindle is assembled to prepare the homologous chromosomes for separation, and there is terminalization of chiasmata, there is repulsion between the bivalents, and these are still connected at specific points. The homologous chromosomes, they start moving towards the equator, and the nuclear membrane also disappears by this time. The centrioles, they start migrating to the poles, and spindle fibers begin to form. So by the end of diakinesis, the nucleolus disappears, nuclear membrane also disappears, breaks down, and these are the major events that take place during prophase 1, which has been divided into 5 substages, and each substage, the events that take place, that occur, I have explained them to you and briefed them as points with diagrams. So it is essential, students, that you make your own notes, write these points, learn these points, because this is an important chapter which you have never done before. Now, after prophase 1, we come to the next stage, which is metaphase 1. Metaphase 1, the characteristic events that occur here are that the bivalent chromosomes, they are aligned at the equatorial plate of the spindle. The arms of the chromatids are pointed inward. The centromeres are pointed towards the poles. The bivalents which were attached at the centromeres now attach to the spindles, and the microtubules from the opposite poles of the spindle attach to the pair of homologous chromosomes. As you can see in the figure shown here, how spindle attaches to the centromere and how the chromosomes are aligned at the equatorial plate. Then metaphase 1 is followed by anaphase 1, and characteristic events of anaphase 1 are that the spindle fibers pull the whole chromosome. The homologous chromosomes then separate while the sister chromatids remain associated at the centromeres. So there is no splitting of centromere. Separated chromosomes, which are one from each pair, move towards the poles. Shrinking of the spindle fiber takes place. Anaphase 1 is followed by telophase, and telophase, the characteristic events that take place have been listed here. Each pole possesses chromosomes which are composed up of 2 chromatids. Due to crossing over, these chromatids are not genetically identical. So this is the difference that is there in mitosis and meiosis. In meiosis, the chromatids that are there are not genetically identical because crossing over has occurred. And the spindle fibers, they usually disappear and chromatids start uncoiling. They form the chromatin network, and nuclear membrane is formed, nucleolus reappears, and after telophase 1, that is after 2 nuclei are formed, and each of these nuclei have half the number of chromosomes than in the parent cell. There, after this, division of cytoplasm takes place, what we call as cytokinesis. Cytokinesis takes place and there is formation of 2 cells. This is completion of meiosis 1, the first reductional division, what we call as meiosis 1. After this, the cell will enter into So after meiosis 1 is interphase 2, which is also called interkinesis. And interkinesis is the stage between 2 meiotic divisions and is generally of very short duration. And this is different from interphase 1 because here no S phase or no replication of DNA occurs and is generally present in the animal cells only. And interphase 2 is followed by meiosis 2. And like meiosis 1, meiosis 2 also is divided into karyokinesis and cytokinesis. And in karyokinesis, there are 4 substages called prophase 2, metaphase 2, anaphase 2 and telophase 2. And prophase 2, this is much simpler than prophase 1. And now we will be dealing with each of these substages of meiosis 2. Now, as I told you, meiosis 2 is initiated immediately after cytokinesis, even usually before the chromosomes have fully elongated. In contrast to meiosis 1, meiosis 2 is a normal mitotic division, and that is why it is called equational division, because here the chromosome number will remain same. And during prophase 2, nuclear membrane breaks down, nucleoli disappear, and the chromosomes shorten and they thicken and are clearly visible. They again become compact, and each chromosome is made up of 2 sister chromatids. There is appearance of spindle fibers between the centrioles that have started moving towards the opposite poles, and the axis of chromatids are at right angles to the spindle fibers of the first meiotic division. Prophase 2 is followed by metaphase 2. And as you can see in the diagram here for metaphase 2, that the centrioles have moved to the opposite poles. The microtubules from the opposite poles of the spindle get attached to the centromere of the sister chromatids, and the chromosomes, they align at the equatorial plate. The nuclear membrane and nucleoli that had disappeared during prophase 2 are not visible in metaphase 2. Metaphase 2 is followed by anaphase 2, and anaphase 2, this begins with the simultaneous splitting of the centromere of each chromosome which was holding the sister chromatids, and it was holding the sister chromatids, and because of which they move to the opposite poles of the cell. The spindle fibers, they shorten and they pull the centromeres. The centromeres then divide and the chromatids are pulled towards the opposite poles. The separated chromatids now are called chromosomes. So these were the important events that take place in anaphase 2. Anaphase 2 is followed by telophase 2, and in telophase 2 the chromatids which are now called chromosomes, they begin to uncoil, they elongate and they form the chromatin network. The nuclei are formed with the appearance of nuclear membrane and nucleolus, and actually telophase is a stage in which there is a reversal of the events that took place in prophase. It is a reversal of prophase. So here in telophase 2 it is reversal of prophase 2. So you will see that the chromatids form chromatin network, they uncoil, the nuclei are reformed because nuclear membrane and nucleolus, they reappear, and at the end of telophase 2 4 daughter nuclei are formed, and each of these daughter nuclei contain haploid number of chromosomes. And after telophase 2 occurs cytokinesis. Cytokinesis is actually division of the cytoplasm, and because of which cytoplasm surrounds each of these 4 nuclei, and at the end of meiosis 2 there is formation of 4 daughter cells, and each of these daughter cells are haploid in nature. So this was about meiosis 1 and meiosis 2, the 2 successive divisions that have taken place during reductional cell division or meiosis. And meiosis 1, as I told you, is actually that part of meiosis where the chromosome number is reduced to half, which is followed by meiosis 2, which is meiosis 2 is called equational division. Why? Because here the chromosome number remains same. It is like mitosis. So I hope both mitosis and meiosis are absolutely clear in your minds now. So students, I would once again say, please make your notes, write the important points, draw the diagrams and prepare well. Now this much is enough for today's class, and in the next class we will do the importance of meiosis, and we will even see the important differences between the 2 kinds of cell division, that is mitosis and meiosis. So I think I will end today's class here. Take care and good luck to each one of you.